Shinod, for inviting me into this program. I'm standing here with great apprehension. I have not given, I think last month I gave the first talk to an audience of philosophers. This is the second one. And the topic is, so at that time I spoke what I work on, but this is my view of what I work on. Right? So, and it's in no way a view from all of the neuroscientists, which is a very large class of people of varied kinds of investigative methods and thoughts. They may all not agree on. Can you hear? Yeah. Okay. Can you hear now? Okay. Yeah, so everybody may not agree this is what constitutes necessary and sufficiency or the method of gaining knowledge in the field of neuroscience. So as Shinod was saying, we work with insects and they are quite far away from human beings. So we are stretching what we observe quite a bit to suit our explanation for how that will contribute to the human understanding human cognition. So I would so the so neuroscience in general, as I have written in the abstract, is trying to understand how human cognition, whether it's human or not human, and perceptions, behavior, all this arises out of bodily material, and quite often nervous system or brain, and even in unicellular organism which does not have a brain or a nervous system, behaviors arises out of configurations and changes in configurations in the body, right? So the first person I think who really connected the brain to perception is, I think, at least this is my understanding, is that Al-Haytham in the 11th century. And so all that insight for him came from his lot of, he was probably the very first experimental neuroscientist. So he was working with all kinds of stuff related to perception and brain, both anatomy and behavior and physics, right? So here he is seen to be demonstrating camera, right, pinhole camera. And this is a picture from his book, 11th century picture, right? So he does anatomy and then he sees these eyeballs, and you can make out. And then there are these two paths which are going into the brain. So he finds this, and he also finds this crystalline pathway for the light to enter, and the light is sort of preserving the neighborhoods in the outside world while it is reaching the eye. So he doesn't get to know, get to the point that there are neurons or any such thing. So he, but he concludes that something on the surface of the eye. He doesn't say retina or the eye actually collects information, preserving the neighborhoods. Like if two things in the outside world are nearby, what is inside is also nearby. But then he says, so in the camera obscura, of course, it is inverted. The image is inverted on the screens, but he maintains that okay, something must be happening inside which brings it upright again. But the interesting thing is that though he tells that about the inside of the eye, he tells that eventually, because this he observes the optic nerve and the optic chiasm, and which is sort of twisted, so he concludes that, oh, it cannot be maintaining the upright path all through. So then that's the first I think the instance when somebody says that it doesn't have to maintain the physical aspects as such while it's going into the brain. It may be changing that, transforming that in representation somehow. That in some sense is the first evocation of that, I would say. Then of course, the first person to sort of say, okay, there is something to do with electricity in the sense of how behavior arises, is Galvini in the 18th century, when he touches a dissected frog with a piece of metal. He finds that the leg flexes. So, and he was working with static electricity, meaning like the comb rubbing and then you get electricity. But he used a metal in this case, and he actually thinks that electricity is actually generated by the body, and that is somehow causing this reflex initiated by the touching. And Volta at that time, who is studying the metal fluid junctions producing electricity, he tells that no, this is not caused by the electricity produced by the, in this particular instance, but electricity produced by your metal, and they have some argument and so on, and eventually it gets resolved that in some sense both are right, and electricity has to be generated to cause it, and electricity is probably generated in this context by the metal. But then Volta pile experiments, or all the electrochemistry comes into sort of picture, and then that is where the starting of understanding the brain function and the functioning of nervous system in terms of some electrical conduction required comes off. So, and then comes a lot of anatomy work. So people find that microscopy develops, and people look at the brain and nervous tissue and find all these reticular network of cells, right? And so there is a debate whether it's one reticular network or is it a number of units which are net-like, but they are not connected physically, but there's a space between them. And Ramon Cajal in the early 20th century is a person who actually sort of first proposes that they are, from his very extensive microscopy works, that they are separate units, and the neurons are separate units, and their connections are not like single cell unit, but somehow they are connected to each other through some kind of communication means. And he also is also the person who and this is important in some sense, because he's just looking at pictures and pictures of fibers running this way, that way, and just looking at the way they are flowing, and that there is some fiber starting from the eye, and then they are connected and then going inside, continuing what Al Haytham probably started off. And he then says, okay, information direction of flow, that kind of a concept is coming from his work. So all that is the background, and so currently when we look at, so why I'm saying that is that it's a lot of inductive work, right? I mean, far-fetched inductive work you start off with. People, and that's an important part of the in the way neuroscience works, probably other sciences as well, because you don't get the full picture. You are looking at pieces, and then from there you are making up theories which are trying to connect to very far away observations from where you actually did the work, right? So eventually people figure out that whether you look inside a human being or in an insect or a rat, in the brain you see this network of cells, which by now is called neurons by everybody. And they also figure out that a neuron connects to another neuron by these junctions called synapses, right? And so junctions in the sense that there's a gap, and a neuron, a cell sitting at one point, communicates its state to the next neuron by releasing some chemicals. So the chemicals which are diffusing across these gaps are the message that that particular cell is in an excited state or non-excited state, so to say, right? And to tell what an excited state is, inside the cell, if you measure, like if you measure potential difference across a battery in your home, you will measure 1.5 volt. In the same sense, if you put one probe inside the cell and another probe outside the cell and measure the potential across the membrane which forms a cell, then you will measure around minus 65 millivolt, right? And then what we call as excitation in that cell is that potential rising from minus 65 to anywhere above, right? And usually there are, in many cells, there are very concrete steps of excitation, which people call spikes or action potentials. And to illustrate here, you can see here it is sitting at a particular potential, and then these upward fluctuations are there, which are lasting a few milliseconds, 1 or 2 milliseconds, we can say, and they are what are called spikes. And every such spike is causing this release of these chemicals, which tells the next cell that, this previous cell has got an excited state, and it is up to the next cell and the expression of different proteins on that cell to interpret that message. So it can be interpreted as if, okay, oh, that cell is got excited, now I should get inhibited, or it can be interpreted as, oh, that cell has got excited, I should get excited. And that, what it's not thinking, but based on the configuration which is at this junction, that's the way the next cell would get interpret that data. So there is a pre-cell and the post-cell, which would say presynaptic neuron and postsynaptic neuron. This is a very what you would call dogma in some sense in the neuroscience way of looking at how brain functions. There are other kinds of junction between neurons which are called gap junctions, which are directly electrically connecting, and they are generally weak, and we don't really understand why they should be connected like that, because our current understanding of how this network functions implies that this information about a particular cell being excited can be interpreted either way by the postsynaptic cell. So yeah, so that is a basic way in which we look at how material inside the brain is functioning. So to tell it is fairly complex. We don't have to understand this picture, but I'm just telling that there is a presynaptic neuron, there is a postsynaptic neuron, and a lot of molecular machinery is enabling this release of neurotransmitter, the chemicals, and reception of it and interpretation of this in terms of excitation or inhibition. And they are sort of stochastic processes, because of which you can imagine it's a very noisy system. Like it's like shouting in a very crowded room where everybody is speaking, right? So a lot of problem neuroscientists trying to work out is about how in this very noisy system very reliable behaviors can arise. Okay. And so that is one thing. So of course there is also people trying to bridge the gap, so people starting from understanding of different subunits on the cell membrane, different proteins, lipids, chemicals, all that interacting. How can we maybe mathematically or numerically capture the behavior of these interacting events, which then can predict how a single neuron can function? And then using those models put together, can you predict how different neurons come together? What is the function that will come out of that? How they will behave, this collection of neurons connected in a certain way, starting from the way these molecules are arranged on the cell. And we have to admit that currently our understanding of predicting how such a configuration will work from experimental details collected about the subunits, which are all these molecules in a particular circuit, is very dependable, to the extent that people will now can run simulations of this part of the brain. I have collected this data from experiments telling this molecule is there, this is the shape of this neuron, and this is connected to this neuron from various kinds of experiments, and you just simulate that, and then people assume that you don't have to do experiments to verify that, oh, this circuit will function like this. So that part is very well understood these days, so it has been replicated a number of times. So challenge often is in collecting that data required to feed into that model in that sense. Now let us take a very simple system. Okay, so this is the background, and so here is a crayfish. Okay, and if you touch a crayfish will reflexively jump to escape the touch. Right, so it's a very simple reflexive behavior in the behaviorist sense in the early times, where people said, okay, stimulus response, right? And we understand how this particular behavior is instantiated in this system. So here is a sensory neuron, which is a neuron which will get excited when you touch, and that is connected to many such neurons are here, and they will get connected to one particular neuron called lateral giant interneuron. Okay. And then that lateral giant interneuron connects to a motor neuron. So here is a simple reflex. So you touch anywhere here, one of these neurons get activated, it activates LGI, very fast neuron, which will then drive the motor neuron, which causes this reflex. Yeah. No, pointer doesn't work, that's right. Yeah. When you move, you are okay. So is there a stick? Then I can. Okay. Yeah, fine. So yeah, so that is a simple reflex, right? Now, as I said, there is a transduction or a sensing of the touch itself, which takes place at the sensory neuron. So, which is a set of change in these potentials in one particular neuron, right? And to sort of illustrate the fact that it doesn't matter whether it is touch or in any other manner that neuron is activated, it will cause this escape to happen, right? So, it's sufficient for the neuron for this behavior to arise. We only need any of the sensory neurons to get activated. Okay, that's at the sensory neuron level. Now many of you must have used wigs or balms or menthol lozenges and something like that, and you put in your mouth and you blow, you feel cold, right? And I don't know whether all of you know, but that cold sensation is not coming because any temperature change in your body. Okay, it is just happening because, of course, in the body there are these neurons which actually get activated when temperature change, meaning temperature goes from the the body temperature down. But those proteins which are present in that cell, it so happens that can get activated by this chemical in all these things, which is called menthol. Right? Menthol is a chemical present in either the Vicks Kigoli or Vicks balm. Okay? And that can bind to this receptor, which is normally activated only when the temperature goes below your body 37 degree, but they will get activated by menthol. Right now, it is sufficient that those neurons get activated for you to feel cold. Okay, so it doesn't matter how the neuron got activated. This is at a very sensory level, at the very periphery, and that is sufficient for you to feel cold. That is the this is a thing which has been repeatedly shown. It doesn't matter how it got activated. Neurons activation can directly cause perception, right? Eventually. No, so that's simple. These are all, you can say, simple reflexive behavior or perception. But it is also true that the activation in somewhere in the brain, suppose you record the activity, this excitation somewhere in the brain of the an animal, and same stimulus is applied in the external world. So the animal encounters exactly same stimulus, but deeper in the brain, the response caused by the same stimulus can actually be different depending on the context in which the animal sits. So that's the next level of complexity which can be added to a reflexive behavior. So here is a monkey doing through three different tasks. So monkey is always fixated, looking straight at it. It's trained to not look this way, that way. So eye is always at this position. Right now, if you flash a light at a particular angle away from that location, it always will fall on a particular location on the retina repeatedly. So in all the three experiments, light is falling on the same portion of the retina. So in the same sense as touching the crayfish or menthol activation, we are activating a particular set of receptor neurons on the retina in all the three cases. Okay, and we are looking at the responses in a particular neuron in the visual sensitive area in the brain. Okay, and what you observe is that if the monkey is not doing anything, it's been just trained to look at the fixation point, then you will get only very few spikes in the that neuron, very little activation in that neuron. But suppose you ask the monkey, you look there, but as soon as you detect the light, you are supposed to turn towards that with your eye. Okay, if you do that, then you observe that before the monkey makes a turn, you actually get increased activity in the particular neuron, which means the state when it is trying to use that sensory neuron to initiate a behavior, the activity of the neuron is different. And if you give a more serious thing, you keep fixated there, but you go and try and reach and touch the that light when it comes on, even then it has an increased response in that particular area, which implies that same reflexive like circuit, right? Light activity in the brain can actually change based on the context in which the animal is sitting. So this is possible. So that's a sort of next level of complexity people found out in the brain. So, okay, this is a little flashback from this monkey experiment. So, so the behaviorism view is sort of challenged first time by Tolman's experiment, right? So, Tolman's experiment is this. So, there is a chamber there, and a rat is allowed to come into the chamber, and there is that shaped tube through which the rat, if it goes, it will be able to find food at that location G. So rat now, if it's put in to learn that, okay, if I walk through that, I'll get G. So it's a tunnel. It's going through the tunnel and reaching the food. So it will, if it is hungry, it will reliably go and do this. Now, what Tolman does is he introduces exactly similar chamber, but he does not have those kind of shaped tube. You have these radiating radial arms, right, and closes the tube which is going to that shaped uh, uh, tunnel. Right. So if the rat is simply following, okay, I'll go straight, take left, take right, and go, then obviously when it comes out of that tube and see the tube is blocked, it will stop searching, or it will just randomly go through every arm in search of food if it is hungry. But what Tolman showed is that more than by chance a lot, rat would go in the arm which, if you draw from here a straight line, it will go to that location. Right, and if it has to do that, then it should have when it was exploring, it was not taking left turn, right turn, it is doing some kind of what is called path integration. So it knows there is a space, and in this space it is this food, and that space can be reached by multiple paths, just like you would use a map, right? So that's the notion of a map or a model of the world which animal internalizes with experience. So it's not the innate world which a lot of things we know about the world sometimes from birth, but this is a model which this particular rat has developed about that particular environment, and is able to use that map to make a previously unexplored trajectory to reach a particular location. That's just like we would use a map. Like if you have gone this way all the time, the road is blocked, you look at the map, if I go like this, I'll hit here. So this is the first challenge to behaviorism. uh, uh, um, okay, so and from this, Tolman is predicting, okay, there should be some way of computing the map in the brain, right? There no neurons, nothing, it's just positing. So far away leap. Okay, now question is, so you also want to show actually really, is it some kind of thing which happens to a rat only if you train it with food, right? Is it, or is it the case that, like, we go to a new place, we walk around, and then we are familiar with the place, and now suddenly you say you go to that market, though you never bought anything from the market and enjoyed it, okay, I know the market is there, I can know the path to that market. Is that the case with rat, right? So again, Tolman's experiment. So what he does is, there is a set of maze in which the rat can be trained with food or let it explore. Okay, so rat is now getting familiar with this maze, so to say, right? And now, if you train the rat to get food from a particular location, you will see that even rat will learn to get the food. So the time taken for the rat to come out to that exit decreases with the repeated times that rat is encountering this maze. But some of the rats are not given food at that exit or anything. So rat is just exploring around, and usually if you put a rat in a new environment, whichever environment, it will explore around and then it will get bored and sit there or walk around the boundary or something. And if you introduce a new object, immediately it gets curious and comes and explore it. It's called novel object recognition. Now you take the rat which has not been trained with reward, but you now start training that rat with reward. So this is the time course. So at this point, you are taking the rat and now start training with reward. You will see that it will immediately find out the path to get the food after a few trials, which means it is already now familiar, in the sense of created the map of this place without having to have got a reward for that. Yeah, so this is actually so people people want to model why things learn, often you have to posit a reward, right? So the way in the modeling literature, because we are trapped in the cost function reward kind of notions, this is called ludic behavior. So when there is no reward, the exploration and playfulness itself is rewarding, and that can evolve in evolutionary sense, because if you played and you learned about the environment in a later context, it is advantageous, as Tolman demonstrated here, right? So again, Tolman is saying that independent of, it is not something to do with training with a reward, but rat is spontaneously developing a map by familiarizing itself with the environment. Now comes, so this is a prediction, right? There should be a map, right? So comes much later. So this is so people want to figure out where is the map, and there there are experiments in which people know that okay, there are some patients who certain parts of the brain are damaged, have problem in forming memories associated with places and time, right? Which is what happens with the map, places, and this happened in this time in this place. So that one of the structures is hippocampus. So these people are recording from the neurons in the hippocampus and measuring the activity of the hippocampus, right? So the rat is put into a place, and rat is exploring around, and then you are measuring the activity of these neurons. And they what they observe is this: when the rat is walking around, there is a neuron which would fire only when the rat is here. It doesn't matter after it has got familiar with the environment. Okay, whenever the rat is in that location, it doesn't matter that looking up, down, or not. It will fire whenever it is in that location. Yet another neuron will fire when it is in this location. So if you take all the neurons which has fired in this rat exploration, you will find that those neurons tile this space, right? So that's then, so they immediately connect it to Tolman's map, right? So here is a system which actually recognizes where the rat is, and not trained for anything. You introduce a rat into a novel environment, it will explore around, and then eventually, so initially when you introduce, you will see initially there is no stable map. After exploration, this stable map develops, and now you take the rat out, and next day you bring in and put in the same environment. When meaning, oh, I know the fan is there, there's a picture there, some table is there, that's all, I know this is the environment. You introduce the rat back into that environment, exactly same map immediately sets in, right? So it doesn't require time then to learn the arena. You take the rat and put in a new arena, which looks different, like a different room. Then again, it has to train for that environment. Now you take, so this is a rat which has two maps, right? Now you take that rat and put it back into the first environment. Immediately the map sets in for that environment, which means it is not the case that next environment erases the previous map. So multiple maps are there, and from the context, animal is able to recognize it is in this environment, and then operate the map on that environment. Yeah, so that is so you can, the leap made by Tolman actually is validated by experiment much many years later. And so this is a kind of so that rat is actually having this particular neuron is active here, and in the there are other areas of the brain where actually it is one neuron is not firing at a particular location, but it fires at regular location on a grid, as if it is forming a graph, graph paper kind of coordinate system. Okay, so many such structures are there which eventually leads to place cells, or these map representing cells in the brain of the so if this is so, then there's somebody is asking, okay, if the hippocampus is so important and many place fields are happening, then for a person who is actually having many places remembered, a lot of details, maybe the hippocampus is larger, right? And so they do this experiment with the London taxi drivers. So this group, Eleanor, this, I think, the PhD student who did that work. So she measured the hippocampal size using MRI technique in people who do taxi cab driving and not. So they find that hippocampus is larger in the people who drive taxi cab in London. Yeah. So that's the notion of, okay, this behavior is there, map should be there. But what's the evidence that that activity is indeed possibly causing this perception of I being in a particular location, right? And that is a train of work which comes from Penfield. You might have seen the Penfield map, homunculus with our somatosensory areas proportionally represented as a human being. So these experiments were created by stimulating the somatosensory cortex. So when you touch the thumb, there is a lot of area representing thumb, so thumb is drawn big, and so on. Okay, so the point is, when people have to undergo surgery, brain surgery, and often to remove certain parts which are having tumor, or certain areas which are having epileptic or source of starting of epilepsy, some focal epilepsy, you need to figure out which areas to be cut out. So roughly, okay, from other experiments somewhere here, but individual persons vary where exactly these different maps and cognitive facilities are located, though roughly we know that. So what the surgeons do is they go and open in this area, and then they figure out, okay, is this to do with speech, is it to do with sensation of touch on the hand, and how do they do that? So the patient is not in anesthesia, general anesthesia, it is local anesthesia, because brain does not have pain sensors. So if you actually take brain and cut the brain itself, you will not feel pain, you will feel other things, right? So here, for example, if you mildly activate, say, somatosensory cortex corresponding to the arm, then the patient may say, oh, I'm feeling somebody is touching my arm, right? So there's a causal factor involved in this subset of neuron getting activated, making the person feel that, okay, I'm feeling the sensation of touch. Or the person will be asked to speak, and then you touch the area which is speech generating. Then if the speech gets stopped and the person get confused, cannot continue the speech, you understand, okay, that area is important for speech, so we should not mess with it. Same with comprehension of speech and so on. Okay, so this is Penfield stimulation, which is the first indication that it is sufficient to cause change in the activity of the certain areas to cause change in perception and performance, right? Yeah, so from all this, if you were to sort of take out what does all this tell us, and what neuroscientists assume from all this experiment is that if a behavior or a perception is different, meaning so some context is there and you find that, okay, this is different from that. Okay, so I ask, is this red paper, and are these two colors same? And if the person detects that these are different, then something measurable inside the organism should be different. This is a very strong assumption which we use. So there is a very strong material measurable stuff inside the brain which is different when perception or behavior is different. So that is a strong assumption, this premise with which we work. So that's about fixed, okay, different perception or different behavior, different activity. Now about learning and memory, right? So everybody knows about the Pavlov's dog experiment, right? So you ring a bell, give food, you see the dog salivate, and thereafter you do multiple times, thereafter you ring the bell, even without the food, the dog salivate. So you think that often the experiment is presented as, okay, the dog learns to associate bell with the reward, right? So the dog has the capacity to learn. But actually, when I teach, I tell them, actually that's not the real inference of this experiment, because anybody who has seen dogs know that dog has the capacity to learn, right? So it's very obvious that so you don't need Pavlov's experiment to show that the dog learns. But it really tells us something else, which is that the dog can hear the bell, right? That's really what it tells you. And why it's important, because when we do experiments with animals, right, we have to first figure out actually the animal can perceive it, right? So unless, how do we investigate whether a particular perception is happening in the animal? We need to have the differential report from the animal, right? And so though we are studying learning and memory using these kind of classical conditioning methods, actually it is often used as a tool to okay, figure out whether animal is differentially sensing two different stimuli. Oh, this bell sound is different from that bell sound. This color is different from that color. This order is different from that order, because one of them you reward, other one you don't reward. Then which one salivates to? Then you know, okay, it can sense that, right? So and so on. Okay. Of course, you can also test whether without reward, whether it has, when it, like that the rat which exploded and figured out the map, whether if it dog simply listened to the bell, it heard the bell, and such experiments are also performed in a slightly circuitous way. Okay. So here is a bee which we work with, which also does the same kind of association, but in this case what we give is, we give a bee sitting there, it has got a proboscis. If you give sugar to the bee will extend this proboscis to drink the sugar. Okay. Now, if you give a little puff of some order, say hexanol or some floral order, we will not extend its proboscis, but you give sugar, it will extend the proboscis. So you give order, give sugar. So order, extend proboscis. You do this for 2-3 times. Thereafter, you give that order, we will extend proboscis even if you don't give sugar. So we really quickly learns this. But if you give a different order, we will not extend proboscis. So it is specifically associating this particular order with reward. You can do it with color or other stimuli. Okay, so they are very fast learning. Okay, so this is the rate at which the bee is learning. So within 3 trials, 80% of the bees would make this association. So now these kind of experiments tells us something else, from which we make some assumption about the internal function of the brain, which is that if the behavior or perception has changed for the same situation or context or stimulus, then something measurable has changed inside the brain. So we are looking for what has changed inside the brain because behavior or perception has changed. Okay, so these are the two premises with which we always neuroscientists work, trying to connect brain with behavior. Different perception, different measurement. Change in perception, change in measurement, right? So that is the as I said, people try to model all this at very nitty-gritty details. Okay, so when we try to do that, this network of neurons, as I said, they have to communicate this information across many synapses, very noisy system. So a big challenge is, how is it that this system is able to reliably perform despite all these fluctuations? Okay. Yeah, so that is just trying to tell that noise which is entering the system can get amplified because of feedback, and yet it is reliable. So it's a big mystery. Another principle which we often use in looking at systems, and often looking at systems to draw what you call extend our conclusion to other organisms, is the fact that the organisms have evolved, right, in a genetic material evolution, and so there is selection, and so these are systems which are evolved in this world. So both are considered. It's not just about evolution. Evolution implies that previously available genetic information is modified, but some of them may not be modified, so conserved, right? The other is that doesn't matter how the genetic manipulation, genetic structure is, and how the brain implements those functions, but organism has to function in the same world, whether it is me or whether it is a bee, right? So there are two things which comes out. When we see, okay, this protein or something like is conserved across organisms, okay, and if I find that this particular machinery comes into play in learning and memory in bee, then because they have evolved from common ancestor, maybe similar functions are implemented by similar set of proteins in other organisms. So studying this may let us conclude about that organism, which is not what we study. So that's how we are able to extend some of these inferences, like for example, how the neuron functions was studied in squid, a piece of squid axon, and the equations governing or at least the structure of the equation governing the neuron in the squid is exactly same as what we now know in human beings. So really, literally translatable to that. The other thing is that it is nothing to necessarily do with genes. It is completely to do with the commonality of the environment, right? So, for example, suppose you are to process olfactory stimuli, right? Olfactory stimuli has a certain physical structure and nature to its in presence in the around the animal. And suppose you as an engineer you were to design something to find out which order is present, what's the concentration, how fast it is arriving, then you may come up with an algorithm and a kind of sensor to do that, right? And why? Because stimulus is like that, nature of the stimulus. So because of which, even though evolutionarily they may not have had a common ancestor which had olfactory sensing ability, evolution, the environment, the nature of the stimuli constrain the process to be a certain thing. So you understand the process taking place in, say, Drosophila, fruit fly, and because it is dealing with olfaction, it has to do this kind of processing, and this is how it is doing the processing. We can look for that processing in dog, and then people have shown that, though in this case, particular case, olfactory circuits have evolved in dog and insects completely independently. They don't have a common ancestor which has the machinery for this. But if you look at how the olfactory circuit looks in the vertebrate and the insect, and look at how the neurons are connected, right, starting from the sensory or neuron up to wiring up to the cortex, whatever you have learned from Drosophila, you would be able to translate into the human beings. That receptors may change and all that, but the architecture and the computation remains the same, completely governed by the nature of the stimulus it has to process. So evolutionary convergence and the what you call evolutionary conservation, right? Conservation of the genes, convergence due to environment. So that is a strong constraint which often can be misleading sometimes, but as a first leap of inference, it is a good measure. Now coming to necessary and sufficiency business in this particular reflex, right? So whenever we talk of necessity and sufficiency in the it is nothing to do with general necessity and sufficiency. For example, suppose I say this neuron inside the crayfish LGI. Okay, if you look at the textbook, it would say necessity and sufficiency of LGI for mediating escape behavior, right? So it is not that you take LGI and put in a petri dish, it is sufficient to cause that crayfish to behave, right? So necessity and sufficiency is completely in the context that that neuron is sitting inside that animal connected in the way, right, in the way it is. So for example, in this particular case, I want to test the necessity and sufficiency of LGI in mediating the behavior. What I would do, necessity, how do I check that? Okay, if I ablate LGI or inactivate LGI, this particular neuron, whether crayfish will still have escape response. And if you say that it does not, then you know it's necessary for having escape behavior. How is sufficiency shown? You activate this neuron without activating the sensory neuron, okay, and then show that this particular neuron, every time you activate, crayfish will escape. Very simple system, but necessity and sufficiency of LGA is claimed only in the sense that in this system, LGA is sitting there like that, everything connected like that, LGI is necessary and sufficient for causing this behavior. A little more complex system which we saw earlier. Here the rat is forming these maps of the environment, and then you can ask the question, okay, these neurons which are very deep in the brain, far away from anywhere in the sensory world, actually very far. It is the limbic system which is gets receiving input no way directly from any of the sensory system. Very highly processed input it gets. That's why it does not change. It's invariant to where the rat is looking and so on. Okay. Now you want to check whether these neurons are necessary for forming this. So there are various tools by which you can inactivate those neurons and show that if you inactivate those neurons, animals do not form place cells and form the map. So it's easy enough to show necessity of these neurons in this context. But what would be required to show sufficiency of this? When I say sufficiency in this context, it is this. So the notion of the map is that if these neurons are activated, that is rat's internal information that it is here, I am here, particular neurons are active, that is what is used by rest of the brain to infer that the rat, I am here, right? So which means if I am actually there and if you want to make me feel I am here, I should be able to activate the subset of neurons which were active here, and then I should feel that I am here even though I am there. That is the sense of sufficiency in this case. Now, until though all these nice correlates existed until 5 years or so back, we didn't have sufficiency proof for this. Now we have sufficiency proof for this. So there are tools by which you can actually, when animal is first getting familiar with an environment, you can label those neurons which are activated only in that environment, meaning some way of chemically marking them, right? And they're chemically marked in a way that later if you shine a particular light on this entire area, only those marked neurons will get activated. It's a very complex genetic tools developed to do this. Now what you do is you are training a rat to be in a particular context A. Rat is familiar with that context and nothing happens to a rat. But same rat is put in a context B, and in that it is trained with either food or in this particular case some shock. If it is given shock in that environment, it will freeze. Thereafter, if you pairing, if you take the rat and put in the context B, even without any shock, it will freeze because it recognizes, oh, this is context B, I am going to get shock, freeze. In the context A, no freezing, right? But you have marked the neurons which were active in B by using this genetic technique. Now you take the rat and put it in context A, and then you shine blue light into the brain. Rat is not seeing any blue light. It is directly through an optic fiber activating the area in which that tagged neurons are there. And then you observe that rat freezes in A and as if it is in B, right? So that is sort of sufficiency of activation of the subset causing the rat to feel as if it is in a different context, right? So this is the sufficiency kind of question we are asking. In a rat, that particular rat, that neuron connected in the same way, it is sufficient, activation of that is sufficient to cause the perception. So people have recorded these neurons which are active on a long, say it's running on a tunnel all the time, okay, in a tunnel, and as we said, there will be different neurons activated at different times, right? And you might have had dreams. Some of the dreams people recall something happened connected to what happened in the daytime, or maybe also mixed up many things, right? Also, there is this notion that if you disrupt the sleep, not notion, it's been shown that if you disrupt the sleep after an experience, that experience, which is an episode or event which you had in the just before the sleep, that cannot get consolidated, and you tend to forget it if you don't sleep. Okay, so you can disturb that, do this experiment. So people think that, okay, there is a replay of these events which took place in the previous day or before the sleep to consolidate this memory, right? And so that's a positing that, oh, there is a replay happening, right, from this observation that disrupting the sleep, the episodes are forgotten. Now, in this case, what you observe is the rat is running on this track, and at some time rat is going to either sit still or maybe it will sleep, right? And if you observe, they observe the same set of neurons while sleeping after it has run to a particular environment. And what you observe is that the set of neurons which were activated in the track, in the sequence it would, as it went through the experience of running along the track, will replay in exactly same sequence during the sleep, but very rapidly. So you might have heard stories of sleep, but the dreams are very fast. The hours of dreams are in very compressed form. So people think that though these are not dreams, these are exact replays of events which happened before, but dreams are recombinations of this. This is now a prediction, right? So we don't yet have any exact verification for that. So given this replay observed in the resting and sleeping rat, replay of the experience as represented by this neurons, people think that dreams are mishmash of these replays. Now, the another concept which we, because of evolution, is that, okay, the notion of purpose and optimality, right? So, okay, there is a feature, the animal is supposed to sense that. Then if, because animal has evolved, whatever thing it has developed to sense or feel or do motor action should be in some sense optimal for that situation, right? So here is an example of such a reflex. So if a gull brings food with its beak to the nest, the chicks would open its mouth, right? So whenever gull's mouth comes, chicks open the mouth, and you can measure how much, how effectively this gull's beak can open the chick's mouth, right? So you can actually, the original gull, a standard model which looks like the gull's head, or different variations of that. You can see how effective are each of these visual stimuli in evoking this uh, opening of the chick's mouth, right? And you observe, okay, some of them are very effective, right? A natural one has very high, and so that's similar. Natural and a realistic standard model. Looks like the head alone without beak does not cause. Beak alone seems to be also somewhat similar to that. Okay. Now you take a pencil and put rubber red tip color on it, and then you extend. That is more effective than the natural beak. So this optimality is not necessarily there, because selection has taken place only for the set of stimuli it might have naturally encountered. So if there is a what's called Bayesian, in the sense the statistics of the stimulus which it has encountered over evolutionary time scale, and if that pencil was never encountered, there was no selection out of detecting that. So whatever mechanism was trying to detect that can actually cause an even enhanced response for a stimuli which it never has encountered, which is what happens to us during uh, optical illusions or different illusions. Those are stimuli which you would naturally not encounter, but you are synthetically creating those stimuli, which are now fed to this system, which is actually using the statistics of what it usually encounters in the world, and you would get illusory perception. So these are illusions that chick is having about what is it. It is a very strong illusion, you would say. Then there is in the when you go to the higher areas of the brain, where it is not reflex, not just maps, we are talking about general capacities in human beings or primates. Okay, when you come to the general capacities, there is a notion of shared resource. Okay, and what's the time like? Yeah, I have like two more slides, I think. Huh? Okay, I will sort of stop here. So the Yeah. So what do I mean by functionality, capacity, and modularity, right? So here is a typical experiment people do, kind of experiment people do to figure out capacity, and it also demonstrates notion of modularity. And what do I mean by that? The task is this: a person is asked to look at the screen, and first a letter, two numbers 9 and 6 is encountered, and the subject is asked to, you have to keep remembering that, rehearsing that number, because at the end of this task you have to write it down. And then some dots appear, and then each of those locations some objects appear. It may be colors, different colors, or different objects, and so on. Okay, it's not making important for this discussion. And then there is that vanishes and the dots appear, and then a particular location is indicated where you should have remembered, where, I mean, you have to remember all of them. You are asked to remember the entire scene also while you are rehearsing that 9 6 9 6, right? And then you are present with two stimuli, one of these, and you are supposed to say it was present or not present in that location, right? So you are, and you can increase the number of such objects on the screen, and so if you can, at some point you will break down, right? If you two objects, easy. Three objects, okay, four objects, five objects, maybe after five objects you are not able to anymore uh, keep any more objects in your, so, and like that you find that's a notion of capacity, right? So you can only keep so many objects in the, yeah. You may stretch it a bit by, here for example they are giving longer time, then you can stretch it a bit. So there is a notion of capacity in the general computation sense of the brain. So these are now not the limbic system. We are looking at the prefrontal cortex and cortical areas where general capacities are there. And what do I mean by modularity here? Is the fact that you you were able to do that 9 plus 6 simultaneously. Okay, and so you were given in this particular experiment, you were asked to do that because you don't want the person to use verbal descriptions in your mind to keep information about the picture. You wanted to use the pictorial memory itself. So you are assuming that these pictorial and the verbal things are separate, and if you occupy the verbal capacity, you can measure the visual capacity independently, and there is a capacity, right? So this is a notion which is um, uh, used when you are coming to studying the subsystems in the brain which are talking about general capacities. Maybe I should sort of stop here and take questions, and if there are questions which are leading, I will go.